Thank you so much. Let me do a little housekeeping here for those of you who have just joined us. Thank you so much for joining us for this Gain webinar today. We've got a wonderful analyst with us, which Steve is going to give the introductions. And so without further ado, I'll introduce you to Steven Saltzstein, the CEO and founder of Force Family Office. Take it away, Steve.
Thanks, Charlotte. And thank you, everyone, for joining us today. We greatly appreciate you taking time out of your day to be with us. We have a fantastic symposium with two of the most qualified folks in biotech and Parkinson's and protein processing. And to begin with, John, do you mind just giving 1 minute, telling us a little bit about yourself and your firm and giving folks some background?
Sure. So I'm a life sciences analyst at Zacks Small Cap Research and cover a number of companies in the small cap life sciences space. I have a background in doing healthcare consulting. Was also on the buy side for a number of years, running portfolios and functioning as an analyst covering healthcare there as well.
Great. You're being modest. That's all I'll say, but that will come out in this discussion. And Matthias, please, tell us about yourself and Gain and all of the amazing things you're doing in Parkinson's and folded protein processing, etc.
Absolutely. Thank you, Stephen. I think we'll leave the substance of you just summarize what we're going to talk about for the rest of the time we have. So I'll just a quick introduction to my person. So I'm Matthias Adler. I'm the CEO at Gain Therapeutics. Been with the company for a bit more than 2 years now, but have spent essentially the entirety of my professional career in pharma and biotech. So that's almost 30 years now. So it's quite a bit of time. So I've seen a lot of ups and downs and cycles in the markets as well, which is obviously very topical to where we are today in biotech in terms of the public markets. But we've had a really exciting program here at Gain, the lead program in Parkinson's disease, which has really the potential to be a breakthrough therapy that can slow or even stop the disease progression. And that's going to be really important for us to move that forward, and happy to be here today with you and John to talk about Gain and all the other cool stuff we're doing.
Okay, super. So I have demoted myself, and I am just going to step in occasionally to interpret terms that might be a little bit scientific for the audience, if that occasionally happens. And John, I'm going to let you take it away. If that's all right with you.
All right. Thanks, Stephen. I appreciate it. And, we at Zacks have talked to Gain on occasion, getting more familiar with the company. And I myself focus on the neurodegenerative space. And also one of the interesting things about the company is that they have a computational platform for identifying certain features of folded proteins or misfolded proteins. And their lead candidate and their lead program is focused on Parkinson's disease. And Matthias, I wanted to see if you could share with us some details on that candidate and how it's different from other approaches, in the clinic for Parkinson's. How do you differentiate it?
Absolutely, and thank you for being here. Look forward to the conversation with you, John. So just taking a step back in terms of just giving the audience an understanding of Parkinson's disease, because it's important to also to appreciate what we can do with our drug candidate. So Parkinson's disease is obviously a neurodegenerative disease, and the symptoms of Parkinson's disease occur when brain cells that produce dopamine, which is a neurotransmitter, when these brain cells die, there is a drop of dopamine levels in the brain, and then the brain cells don't communicate well with each other anymore. And that ultimately leads to the development of these Parkinson's disease symptoms, which are, the well-known symptom is like the tremor, so the lack of control of motor activity, muscle stiffness, slowing of movement, slurred speech. And these symptoms are barely noticeable at the beginning, but it's a progressive disease. And so as these dopamine-producing brain cells continue to die, there's less and less dopamine available. And so these symptoms get worse and worse over a period of time. And ultimately also in some patients, it leads to cognitive decline. And the current treatments that are available for Parkinson's disease, They are addressing the symptoms. So there's some dopamine replacement therapy. So you're adding dopamine artificially into the brain, and that allows the management of the symptoms. And it looks like there's enough dopamine in the brain, but eventually,
wears out.
Exactly, it wears out, and the therapies stop working, and then these patients fall off the cliff on the back end. And what we're doing at Gain is actually, we're not trying to manage the symptoms or deal with the symptoms. We're actually addressing the root cause of the disease. And our goal is to slow or stop the death of these dopamine producing cells, so we have the ability to slow or even stop the progression of the disease itself.
Yeah, and as an analyst, I'm always looking at a company in through the lens of the opportunity for profits and gain and things like that. Right. And I think I'll point out a few stats. There are about 1 million people with Parkinson's disease in the United States, and I think about 8 to 10 million around the globe. So it's a large market, and as you said, Matthias, there are no other disease-modifying therapies out there to address it. So it seems pretty attractive from that point of view. What are some of the other reasons that you selected Parkinson's as your lead candidate and lead program?
Right. So I think more specifically, we're looking actually at, as our lead indication, we're looking at GBA1 Parkinson's disease. And the reason that's the disease in these patients occurs because of a mutation of the GBA1 gene. Mm-hmm. And the reason that is interesting is because that neurodegenerative diseases generally are known to be very heterogeneous. So the origin of the disease, and in Parkinson's, the reason for the death of dopaminergic neurons can be coming from different, can be due to different factors. And in GBA1 Parkinson's, it's actually the largest a genetic risk factor for the development of Parkinson's disease, affecting up to 15% of Parkinson's patients. The link between that genetic mutation and the development of Parkinson's disease symptoms ties very directly into the mechanism of action of our drug. And so it makes it a very direct, it reduces the risk, the clinical development risk, because we know what our drug does, which is really restoring the function of a very important enzyme that's important to maintain cell health in brain cells, which is called GCase. And GCase is expressed by the GBA1 gene. So you have the GBA1 gene creating the GCase enzyme. If there's a mutation of the GBA1 gene, the GCase enzyme is misfolded and dysfunctional, doesn't work properly, and that triggers a disease cascade that ultimately leads to the death of dopaminergic neurons.
So if you can get in there at the beginning of that cascade, you can potentially address the disease.
Precisely, and we can address the disease actually at the beginning, so where that whole process starts right. Developing, right? As opposed to, and that's where many other approaches in this field have failed, is if you're coming on the back end, you're trying to correct the problem once it has already happened, then you're probably falling too short and don't have the profound impact that we have seen in our preclinical models, which, based on the data that we have, our program is the conviction to say that we have a best-in-class profile with our drug in terms of the impact on that entire disease cascade and repairing that disease cascade, restoring cell health, increasing survival of dopaminergic neurons, increasing dopamine levels in the brains of the animal models, and improving the motor function that we have seen in these animal models. So we're fixing this entire problem by fixing the problem where at the beginning where it starts.
Yeah. John, do you mind if I ask Matthias a quick question? Because I'm willing to bet the audience is wondering. Matthias, are the symptoms for GBA1 the same as regular Parkinson's, or yeah?
That's a great question. It's actually another reason, in addition to having that clear link to the mechanism of action. The other reason why this is an interesting indication for us is that GBA1 Parkinson's, so that genetic form of Parkinson's disease, these patients actually have a more severe form of Parkinson's in that they have an earlier onset of the disease. So it starts earlier in their lives. They progress faster on the disease, and they also have more often than not cognitive decline going alongside disease progression. whereas in normal non-genetic Parkinson's patients, cognitive decline happens if it happens at the very back end of the disease. So this is a more severe form. It allows us, because it's a faster progressing disease in these patients, to pull in the development timeframes in the clinical studies that we're going to be running in patients, because we're going to be able to see a difference between placebo and the treatment arm more quickly. And it is an area of very high unmet medical need. And so, I mean, ever since we started clinical studies, now we're in a phase 1 clinical trial, we're getting inundated with calls and emails from patients asking us for ways how they can get into the clinical trial, clearly demonstrating that there's a very profound need and very
that must really help with the enrollment process, I guess, right, Matthias? Just all that.
Exactly. So I do believe, and it's again different from just treating symptoms. I think these patients, when we're starting to enroll patients in the clinical trials, then we're going to see, I expect, a very high interest, because it's a very differentiated approach. So
you mentioned GBA1 gene, but that's also part of another disease, a rare disease called Gaucher disease.
Yes.
Are there any synergies to working on both of these indications at once?
Very much so. I mean, it's actually interesting. You're asking the question. Our lead program that we're now developing as a lead indication. I keep saying lead indication because there's other applications. So the lead indication in GBA1 Parkinson's disease. That program started out as a Gaucher disease program.
Oh, okay.
So we knew that GBA1 mutations cause Gaucher disease. This is even a more severe form than GBA1 Parkinson's, because this starts in early childhood. So this is a childhood rare disease caused by that genetic mutation with the same mechanism of action, with a misfolding of GCase that then needs to be restored with our drug. So we have the ability not only to look at GBA1 Parkinson's disease, but also Gaucher disease with the same molecule. We have additional data and new research coming out that directly links the mechanism of action that we're pursuing with even additional indications, neurodegenerative disease indications like Alzheimer's disease, dementia with Lewy body, multiple system atrophy. So there's a range of additional neurodegenerative diseases that are accessible based on the mechanism of action that we have with our drug. And so this product is really it's a pipeline in a molecule.
Are there any modifications that you need to make to your candidate to use it for Gaucher, or is it the same?
No, it's exactly the same molecule. Okay. Because what we're fixing with the molecule is the functioning of GCase, so restoring proper folding of a misfolded GCase, restoring GCase function, and that reads across that same mechanism. That restoring GCase function has that effect of restoring cell health and improving cell survival, and that ultimately is what you're trying to get to as a disease-modifying therapy in neurodegenerative diseases, is improving the survival of brain cells, and that's basically what our drug does.
Right. So Gain has worked with a lot of recognized leaders in the Parkinson space, and I think probably the one that everyone would recognize is the Michael J. Fox Foundation.
Yes.
But there are some others beyond that. Share with us how it is working with those, how they help you, and how it benefits the whole, I guess, ecosystem of sponsor and innovator and the organizations that represent the patients.
Absolutely. Yeah, we're actually very grateful for the external validation that we have received for our program through these competitive grant funding processes. So we've received an initial grant early on from the Michael J. Fox Foundation when we pitched the concept of what we were trying to do with the drug, and they thought that it's really intriguing, and so they supported the program early on with grant funding. We've also received funding from the Silverstein Foundation for Parkinson's with GBA, and most recently we're able to attract a very significant grant funding from the Swiss Innovation Agency that supports these breakthrough therapeutic development programs. They've provided us with $2.8 million worth of grant funding to advance our clinical stage program. And all of that it shows sort of the evidence that obviously what we're doing is very impactful, very meaningful, and recognized by these groups that, you know, can fund any number of opportunities, but they picked our program. And especially with the Michael J. Fox Foundation, that continues to be very close cooperation. We're participating in their biomarker identification program. So it's a long-term clinical study that we contribute to and are a participant in to develop biomarkers for Parkinson's disease that we then can use on our end as we're looking at patients, when we start treating patients, looking at the biomarkers that have been identified as being showing disease progression, impacting those biomarkers would be an evidence of slowing or stopping disease progression. That's what we're getting out of that relationship with the Michael J. Fox Foundation, in addition to just ongoing support.
And, you know, biomarkers are so important, I think, for a wide variety of reasons. I mean, one of them is it's just very difficult to measure. It just takes a very long time to measure an effect. You know, like I think maybe Alzheimer's is a really good example. Right. So you want to get a sense right away whether or not it's working. So you know, biomarkers have really emerged in recent years as being an important, you know, an important component of developing a drug. What are some of these biomarkers exactly? I think some of us may have heard of neurofilament light, which is used in a number of neurodegenerative diseases. Is that one that you're investigating for Parkinson's?
Yeah. And Matthias, can I just ask, like, before you go into neurofilament light?
Can I just go down the rabbit hole?
Yeah, I prefer Bud Light, but I'm just kidding. But can you just explain what biomarkers are in more? Yeah.
Matthias, you want to do that, or I can do it? Or
let me take a crack, and feel free to jump in.
You'll probably do a better job than I will.
All right. So biomarkers are really you're looking at, on a cellular level, you're looking at the cell of a patient, and you're looking at what the drug does in that cell. And there are proteins that are created in the cell that are either going up if disease progresses and the patient is sick, or they're going down if if your drug has an impact and actually has an impact on the disease, and you start to see a treatment effect. And these are early indications of the potential that the drug might work and ultimately will have impact on the clinical symptoms like the tremors and other things that we're looking at, the cognitive decline that we're looking at in our Parkinson's disease population, which take, 12 to 18 months to actually develop. We're starting to see a difference between placebo and treatment arm in those clinical studies. And with biomarkers, you might see an effect as early as 2 weeks, maybe a month, or even, after 3 months, you should be starting to seeing a difference at the cellular level if the drug does what it needs to do. And the great thing about our program is that we have a whole range of biomarkers that we can look at, because we are starting at the beginning of treating the disease, at the beginning of that disease cascade. We can look at a range of different toxic materials that are built up in a sick cell, in a diseased cell, that we are actually reducing these, and we have shown that preclinically we're reducing these toxic materials in the cell in animal studies. We can also look at neuroinflammation biomarkers, neuroinflammation being a driver of neurodegenerative diseases. We have shown a reduction of neuroinflammation in our preclinical animal models. And we can look at neurofilament light chain, and we're particularly excited about that biomarker because neurofilament light chain is a protein that forms a part of brain cells. And if you have a neurodegenerative disease, meaning your brain cells are dying, that protein is released from the brain and goes into the bloodstream. So if you have high NFL levels in your blood, it means that you have a neurodegenerative disease process going on.
And it's a relatively, one thing I point out is that sometimes collecting biomarkers, just getting them is one of the hardest things to do. And I think you can do a blood test for NFL, right?
Precisely.
And that's opposed to doing like a spinal tap, I think, which may be required for, nobody wants a spinal tap. But I mean, that's another point that I think is really important with that biomarker specifically, is that you can measure it just with a blood test.
Right. So we can measure it in blood, and the exciting element for us is that in animal models, we have seen a very dramatic reduction of NFL levels compared to sick animals after they have been treated with our drug. So we have seen a very significant decrease. And if we're able to demonstrate that in patients, it's very direct evidence that we have a neuroprotective effect, so that we're stopping these brain cells, these dopamine producing brain cells from dying, which means that there is conviction, even more so than we have today, that we have the ability to slow or stop the progression of Parkinson's disease.
Yeah.
Thank you.
Well, biomarkers is one of the kind of the leading interesting areas in neurodegenerative disease. Another one that's more broad, I guess, than that in life sciences is artificial intelligence. And I think over the last year, with ChatGPT being available for the masses, we can kind of get a sense of what AI does. I mean, AI has been used in life sciences for a very long time, right? And it's just getting better. But I don't think people really saw that so much. But now when we have it on our own desktop, it's just a lot more clear what it's doing and how it works. And you guys have a computational platform that uses AI to identify, I think, allosteric binding sites on misfolded proteins. I hope I got that right. And just one other thing I wanted to say on that, people may have heard of Google's AlphaFold, which I think tries to do something similar, or well, it tries to predict protein folding. So, I'm just trying to tie in kind of some of the things that are out there that people may have heard of. Maybe you can compare what you're doing with some of the other platforms that are out there, and how you're using AI to, I guess, save time and save money and create more precise drugs.
Right. Yeah, I mean, Gain really at its core started out as a computational drug discovery company. So with our platform, we have identified all of the products that we are currently pursuing in our drug development pipeline, including the lead program that now has moved into the clinic. So in a way, it's actually one of the few computational platforms that has generated a clinical stage program. So in that sense, it's actually a highly validated platform. It's very fast, so it accelerates drug discovery. Instead of spending 2+ years in high throughput screening, we can get to a molecule that can be developed into a drug in less than 3 months. So but significant acceleration. So, and time is money in pharmaceutical development, right? And so we're really accelerating that discovery phase is very important. It's also very versatile in that we can look at any protein in the human body, tying back to AlphaFold, that can predict the 3D structure of any protein in the human body. We can use these AlphaFold structures in our platform to look at these novel allosteric binding sites. And so we're using some AI in that, in terms of using AlphaFold, but at the core, it's really a physics-based approach. So AI, as you will know, requires a lot of pre-existing data to train the models that, to extrapolate from pre-existing data to something else, something new, which makes it really hard if you're trying to find never seen before binding sites on the surface of proteins, because there's no pre-existing data by definition about, these binding sites. And so what we have the ability to do with our physics-based approach is looking at the entire protein surface, measuring binding energy between our molecular probes and the protein surface, and finding these novel binding sites. And we recently upgraded the platform to access virtual screening space for chemistry, for new chemical compounds of 50 billion plus compounds that we now can screen against these novel binding sites that we have found, to find new small molecules that can be developed into drugs. And if you're thinking 50 billion is obviously a big number, but if you think of it as if you're screening one compound per second, which is pretty fast, right? To see if a molecule binds onto the protein surface. One compound per second, it takes you 16 centuries to screen 50 billion compounds. And we can do that same process in a handful of days. So it's clearly an incredibly powerful.
It's a lot of computing power.
A lot of computing power that we can harness with our platform, the models that we run on it. Yes.
Okay, great. I thought we'd point out something just in the recent news for Gain. You recently had publication in PLOS One on GMT1 gangliosidosis, and thought you could update us on what that's all about. And it's always nice to get published in a well-recognized journal. And how does that help out the program, that publication?
Right. So GM1 gangliosidosis is a rare lysosomal storage disorder caused by a gene mutation that then leads to a misfolded enzyme. So actually the same theme that we just talked about with respect to Gaucher disease and GBA1 Parkinson's disease. So we're restoring, what we have shown is that we have the ability to restore enzyme function and enable that enzyme then to do its job, which is to break down fats and sugars in the body. If that doesn't happen, it leads to a buildup that ultimately leads to seizures and vision loss and things like that. And so what we have shown in our in that publication is that we have been able to restore the function of that enzyme and start reducing that buildup of these materials that otherwise cause that disease. And so there's a common theme that we have with the lead program, another application, a different protein, but it just provides further evidence that it's not a fluke, right? What we have found in our program, there's actually a very systematic approach that is enabled by our platform that allows us to find these allosteric binding sites on misfolded proteins and then find molecules that can restore protein function.
I thought it might be helpful to explain what an allosteric binding site is in contrast to, the target binding site. What are the choices there in terms of binding sites, and why would you select the allosteric site?
Absolutely. There's essentially two types of categories of binding site. One is the active binding site on a protein, and that's where the protein in a body works when there's like an interaction with another element within the cell. And so there's a natural ligand, a natural binder that occurs in the body that engages with the protein at the active binding site and then triggers either an activation of protein or stops the activity of the protein by binding there. That's just a natural function. And diseases occur, protein-driven diseases occur if that interaction is not working properly, and so the protein gets activated when it shouldn't, or it gets inhibited when it shouldn't. And
so I guess you're oh yeah, go ahead. Sorry.
That's the active binding site, and essentially the allosteric binding site is anywhere else on the protein surface where a small molecule can actually bind and engage with a protein. And our platform is uniquely suited to look at the entirety of that protein surface to find these allosteric binding sites and finding small molecules that engage with the protein on those allosteric binding sites. And these are unique binding sites. They're not shared across a whole range of other proteins like the active binding sites or the sort of protein families that share commonalities. And so allosteric binders typically have less toxicity in terms of their interactions because it's a very unique site that is being identified. Now we have different ways to affect the protein. So we are generally either restoring or disrupting the function of a protein. But that specific mechanism of restoring the function of a misfolded protein, you can only do by binding at an allosteric binding site. Binding to the active binding site wouldn't do that. And so it's a very unique mechanism of action that we're exploiting with our lead program that has been uniquely made possible with the platform that we have at Gain Therapeutics.
Great. Well, Stephen, I think I saw some questions, at least I see some numbers there.
You know what, I have one before we go to audience questions. Matthias, do you mind talking about the total addressable market for your Parkinson's program and then also for your, protein screening technology?
Right.
I just want people to get a sense of the size of both markets.
Right. So the total addressable market in GBA Parkinson's disease is probably about 150,000 patients in the US. We have the ability to expand the program into idiopathic, so non-genetic Parkinson's disease, so that makes it about 1 million patients in the US. We have the ability to expand it into Alzheimer's disease and dementia with Lewy body and other neurodegenerative diseases. So in terms of actual amount and market opportunity, market potential, it ranges from about 1 billion to 1.5 billion for the smallest indication to a multi-billion product opportunity overall.
Okay. Sorry. That's for the actual program. But
yes.
what about for the technology? Or
well, the technology obviously allows us to generate our own pipeline and continue to build new programs essentially at will and very inexpensively. So from that perspective, it's actually a strategic sort of advantage of having the platform in-house at Gain. But it also enables us to, because it's so broadly applicable in any indication, any therapeutic area, it does open up the path to discovery collaborations with larger industry partners, pharma companies who also look to accelerate drug discovery, or maybe have proteins for which they could not find a suitable binding site. With our platform, we can find these binding sites in small molecules that have that therapeutic effect. And so it is the opportunity to tackle the 90% of proteins that so far have been undruggable because they don't have a known binding site. With our platform, we can find it. And so it's an opportunity for us to generate non-dilutive funding through industry collaborations. Now that we have a clinical validation with our lead program of the platform, we expect to actually get that traction with pharma partners who start to take note about the validity and value of the platform that we have.
But do you have any sense of, what it could be worth? Or, I mean, if you're not comfortable saying, that's fine also. What I'm trying to do is give people a sense of, I think today your market cap is 37 million. Your own programs alone, a total addressable market, or somewhere, it seems to me, anywhere between 1 and 2 billion. Plus you have this whole other side of the business.
Right. Well, I mean, clearly there is a sort of dislocation of market evaluation, and so we have actually a nice comparator. We have a company, Merck acquired a company recently, Gain Therapeutics, and they were working actually on a downstream effect of that GCase dysfunction I described before. So we're acting upstream, fixing GCase. They were working downstream on one of the effects of dysfunctional GCase. Preclinical stage, so earlier stage than we are, way earlier stage than we are, way downstream compared to where we're acting with our drug. And the total deal value for the acquisition of Caraway was $610 million. Compared to what you said, our market cap today is about 37 million. So clearly, there is a disconnect between the value that for industry partners and pharma companies see in programs in our space, even at earlier stages compared to where we are, compared to what the public markets recognize in terms of value that we have within Gain, which means that there is obviously significant upside potential. And as we're progressing our lead program that's in the clinic now through various clinical value inflection points over the course of the next 12 plus months, there is an opportunity to start seeing a realization of that this disconnect and an increase in value as we're achieving these value inflection points. Super.
Thank you. Charlotte, take it away.
Thank you. Well, the very first thing I think we should say is that the ticker symbol is GANX. So we want to make sure that we let people know that GANX is the ticker symbol. And then we'll get to the questions. Thank you so much, John and Matthias. What are the initial signs of GBA1?
Initial signs of GBA1 disease. I would complete that question. So the initial signs are, it really starts out with some actually very early, even before the movement symptoms. You start to have a disruption of your sleep cycle. You start to have potentially also constipation as an early sign of GBA1 Parkinson's disease, or Parkinson's disease more generally. And then you start to see tremors that are in one or the other hand that can't be controlled well. And that's of the initial motor symptom that based on which in the past Parkinson's disease has been diagnosed. There are now tools out there that allow for the diagnosis of Parkinson's disease, or the process of that ultimately leads to the motor symptoms of Parkinson's disease. as early as 7 years before these motor symptoms, the visible symptoms occur, by measuring through accelerometry. So there's now programs where you can measure the way you move. A movement it is a movement disorder, so balance, speed of movement, how fast you get up, losing a bit of loss of balance as you walk. That can now be measured in a like a Fitbit or an Apple Watch. You can have a program like that, and up to 7 years before. There are methods now that allow the identification of these very, very early signals that Parkinson's disease might develop, up to 7 years before. And that's, if you're thinking about the drug that we're developing, ultimately that's the type of patient that we want to be treating with our drug, because with our drug, we have the ability to slow or stop the death of brain cells. And the more brain cells you still have, the more complete the function is that you can maintain, either for a prolonged period of time or forever if we're super effective with the drug.
Amazing. Thank you. Why is Parkinson's so deadly?
Well, Parkinson's in and of itself is actually not necessarily deadly. It's highly debilitating over time and will require a significant amount of care. But it's not like cancer, where ultimately it takes over and you die of cancer. People typically die with Parkinson's disease as opposed to of Parkinson's disease. But the effects and the debilitating effects are really devastating. And, almost everybody we talk to in their own family has had somebody or has somebody with Parkinson's disease or another neurodegenerative disease. And you think of in my own family, I have, we've had Parkinson's disease, and it's just in the end, these patients with Parkinson's disease, they cannot care for themselves anymore. It's just the quality of life is really terrible. And if we're able to push that out 10 years or forever with a treatment like ours, it will incredibly improve the human condition, the condition for these patients.
Yes, the effect on everybody, just not the patient themselves. My father-in-law had Lewy bodies, and as you said, he didn't die of that, but it's awful. It's awful to watch. Thank you. Is this the type of Parkinson's that Michael J. Fox has?
I actually don't know if he's done genetic testing. So GBA Parkinson's is obviously the one that is driven by that genetic mutation. Given how early he was diagnosed with it, you could suspect it might be. On the other hand, it hasn't progressed very fast because he still has a lot of ability in terms of if you're sort of seeing him in public appearances. So he has had a really, I guess, also good level of treatment and care throughout his life, and that has allowed to extend that.
Thank you. How many people are enrolled in your phase 1, and what will the endpoints of what will be the endpoints of phase 2?
Yeah. So the phase 1 study that we've started in the 3rd quarter of this year is going on. It's a study in up to 72 healthy volunteers. So we're first looking in healthy volunteers as opposed to patients, and looking for how far. It's a dose escalation study. So we're starting at low dose, a cohort of patients, and then stepping up to the higher dose and so forth. And that process is currently going on and progressing well. So we're on track to complete the study in Q2 of this coming of 24. We're also at this point planning to expand that study and add a patient cohort to that study, where we're treating them patients for 3 months, with the goal to look at the change in the biomarkers in these patients that we have talked about earlier during this conversation, looking at a range of biomarkers and seeing that we can reduce the, say, NFL and other neuroinflammation and other biomarkers that show that the drug actually works. And that study, that arm is studying in the 3rd quarter of this year, and then moving into the end of the year, early 25. And that will be giving us that early indication that the drug actually might work in patients, as we have seen it working in the animal models of Parkinson's disease.
Great. Well, I certainly hope that happens. You have orphan drug status. Do you have any other awards from the FDA?
We have not yet gotten any awards or sort of regulatory, recognition from the FDA, really because we have not started clinical trials in the US. We're doing the phase 1 study in Australia. The reason we're doing that is because there is a good phase 1 infrastructure in Australia to run a phase 1 study. And that exists because the Australian government provides a cash refund of 43.5% of every dollar we're spending in Australia, we're getting as a cash back. So it's not just a tax rebate, it's a cash back to the company. And so as a small biotech company trying to be capital efficient, it's a great place to run the phase 1 study. We're planning then to take the phase 1 data back to the US and file an IND for that, and the phase 2 study that's going to start in 2025, and that's going to be, and the FDA has routinely accepted data generated in Australia for the path that we're pursuing. So this is not invented by Gain. Many other people have pursued that same path for the same reason.
For sure. Thank you. Is there a compassionate use program for your drug?
Not at this time, because we're not yet in patients, and we can look at compassionate use once we start treating patients, once we've passed that initial safety gate, right? We need to demonstrate safety and tolerability in healthy volunteers, knowing up to which dose level we can dose. And at that point, once we get into patients, we can look at compassionate use or enrollment directly into the study that we're actually doing, which is something we'll be looking for.
Can you touch on your financial condition? Will you need to do a capital raise soon, and how much will you need? And why do you think you trade at such a deep discount to the price targets on the street?
Well, in terms of capital requirements, we have at the end of the quarter of the third quarter of this year, we had 12.2 million cash on hand. We subsequently then raised $10 million, $10 plus million, $10.1 million to provide the cash to really run the phase 1 clinical study, including the patient cohort, to generate that value inflection point for our shareholders with that biomarker-based clinical proof of concept that we expect to get to with the money that we have raised. And that was the purpose of the raise. So we are financed through that point from that perspective, and don't have immediate plans to raise more cash through equity funding. We will be looking for further grant funding. Interestingly enough, the Michael J. Fox Foundation that we talked about before, they initially really only funded research programs because there was not much going on in the clinic. But now that many of their programs that they funded in research actually have moved into the clinic, including ours, will be going back. They started to give grants for clinical stage activities. And so we're going back to the Michael J. Fox Foundation early next year and see if we can tap into that foundation money to help support the clinical study as well, which obviously will add to the cash runway.
Great, thank you. What is the average age of someone with GBA1 Parkinson's?
Well, it starts about 5 to 10 years before idiopathic or non-genetic Parkinson's. And so you're looking at sort of mid-50s to early 60s as the onset versus mid-60s to 70, so for the idiopathic.
Thank you. When can we get an update on the rest of the pipeline, i.e. cancer programs?
Well, the cancer programs are ongoing. They're obviously earlier stage, they're research stage programs. And in terms of direct value creation, we see really the lead program as the main driver. And so we're running these programs, but the most of our financial resources that we have, that we've just raised the money for, are obviously allocated and designed to support the clinical program. And so while we're pursuing the earlier stage programs, we have opportunities for all of our earlier stage programs through grant funding. We're progressing one of those programs for alpha-1 antitrypsin deficiency with a grant that we've gotten in Europe and a consortium of collaborators. So again, a capital efficient way to advance early stage programs to more tangible value inflection points. And we're seeking to do the same thing for other programs, including oncology programs, as well as collaborations on the earlier stage programs, so business development activities as well.
Great, thank you. When would you expect to have results on efficacy of your lead compound?
That ties back to that phase 1 plan that we now have and that we recently revamped to add that patient cohort in that phase 1 study, where we're going to be treating them for 3 months and looking at biomarkers. And that will be, clinically speaking, the first evidence that the drug works in patients, as we have seen it working in animal models of Parkinson's disease, looking at biomarkers after a treatment of 3 months. And the benefit of the mechanism of action that we're pursuing is fixing that GCase function at the very beginning of the disease cascade. We can look at a reduction of toxic materials. We can look at look at a reduction of neurofilament light chain that we have looked at. We can look at a reduction of neuroinflammatory biomarkers. All of those tie directly to the mechanism of action that we have with our drug, and tie directly to the effect you can expect to see later on in longer clinical studies when we're looking at movement and cognition in these patients.
Okay. So one more before we have one for John. So get ready, John. One of our attendees is interested to find out if they could get on a list to see if the treatment might apply to them or one of their loved ones.
I think probably best, I mean, for one, right now we're in healthy volunteers, so we're planning to start enrolling patients in that patient cohort in Q3 of this coming year. I mean, it's not that far out. So we, as I mentioned, we often get inquiries from patients and families who have somebody in their family who has Parkinson's. So happy to take that email and follow up then with the person. Yeah.
Thank you so much. We'll get you'll have everybody's email at the end of this, as well as if someone is interested in getting in touch, they can contact us directly. You'll have a calendar email.
Yeah.
Thank you for that. And then now we've got one here for John. John, can you talk about the biotech landscape overall? What's promising news, and what does the M&A landscape look like?
Sure. I think a lot of times people think of the M&A landscape, which you mentioned, is the exit strategy for a lot of smaller cap companies. The small cap companies have enough funds to take a candidate up to the proof of concept stage, and then they offer it out to large pharma, large biotech to take it from there and get it in front of the FDA and get it approved and then commercialize it. So one thing that I look pretty closely at is M&A and how it's been trending. As Matthias mentioned, there was recently a transaction, Gain Therapeutics, which had a very similar profile to Gain, which was acquired for, I think, 610 million of bio bucks. So that's a good sign. We also saw a transaction just yesterday in the GLP-1 space, which is the weight loss space that's been kind of taking over this year, for I think 2.7 billion. Merck acquired a small company, well, not small at 2.7 billion, but they acquired a private company that's working in that space. Year to date, M&A has been pretty strong as well. If you compare it year over year, I think I took put a little note on the screen here. There have been 110 announcements as of Monday, yesterday, valued at just over 213 billion, and that compares to last year where there were 81 transactions worth 88 billion. So you can see it's up quite substantially year over year, both in the number of transactions and the size of the transactions. So that's a good sign. Another thing that I think we can all look at is the XBI. Maybe some investors are familiar with that ETF, which mirrors the performance of the life sciences space. It bottomed out, I think, October 31st, and it's up dramatically since then, maybe 30% or something like that. It's just been on a run for the month of November and into December. So there are a few good signs out there that things are turning. With interest rates going up, I think that was a pretty big hurdle for capital hungry companies to overcome. So since interest rates have come down, and I think a lot of investors have recognized value in some of these lower price, Gain is a great example of that. It's in between 30 and 40 million market cap and has opportunities in, potentially the billions. And then also there was one other point that I wanted to make. We're coming off the bottom, interest rates are down, making capital potentially more available. M&A has increased year over year. So I think these signs all together indicate that now could be a good time. I mean, one thing I said in an interview I did a couple of weeks ago, or maybe a month ago, was that I think in a couple of years, we can look back on the fall of 2023 and realize that probably was a good time to get in, kind of like some other periods in time. So I think that would be my short assessment of kind of the life sciences
landscape
group right now, and kind of what, some of the things I'm looking at and my perspective on it.
Great, thank you so much. Well, that ends the Q&A portion. I'm going to give you guys both a couple of seconds to wrap it up. Matthias, do you have any last words?
Last words? No, thanks everybody for participating in the webinar today. I hope you have gotten some interesting information about Gain and what we're doing. And we're really excited about where we are today, having now turned the corner and becoming a clinical stage company in Q3 of this year, progressing our clinical program with significant value inflection points coming up, for which we are funded and have the money to actually achieve. Having the opportunity really at Gain to develop a drug that can have this profound effect on patients with Parkinson's disease and other neurodegenerative diseases by slowing or stopping disease progression is what everybody's really waiting for and hoping for. And so it's for us, it's an important mission that we have. It's an exciting time for us at Gain. And if you're thinking about it, to John's point, from an investor perspective, having just completed the financing that we needed to do to achieve these value inflection points and still trading at relatively low levels, it's obviously compared to, say, the private market valuations that we have seen through the M&A transactions that John just quoted, there is, I would say, is a significant value dissonance between where we are at currently valued based in the public markets, and I believe where we properly should be valued based on the inherent value of our program. So we look forward to progressing that, achieving these value inflection points, building value within Gain, and seeing that we can get this drug into a position where we're going to be either progressing it into later stage clinical studies or ultimately getting acquired by a larger pharma company to then take that program all the way to the finish line.
Great. John?
I just appreciate everyone joining us today, and hope they got something out of my questions and Matthias's responses. If you'd like to hear more about some of the things that I'm working on and what Zacks is working on, you can follow me on Twitter, and that is @VanJohn10, @VanJohn10, and you can find all my stuff on there. I've also got some other platforms as well, but you can find everything I do on there, and if you'd like to follow, please do.
John, say that one more time, your Twitter handle.
Yes, it's @VanJohn10, VanJohn10.
Super.
Okay. And Steve?
Thanks so much, guys. You did a fantastic job. It was a fascinating discussion. We greatly appreciate it.